it's my great pleasure to introduce Professor Sir John Bell. John doesn't need an introduction. He's Regis Professor of Medicine at Oxford and has held many important offices within the UK. Most recently, he was the author of the Life Science Industrial Strategy and also is leading really the driving force behind the new 5 million cohort, the accelerated disease detection cohort. So John, thank you very much.
Thanks, Munir, very much, and well done on everybody getting here today. I suspect that wasn't always straightforward. This is slightly strange. I'm not a public health doctor, but I do think a lot about public health issues, so this is a slightly orthogonal talk for me. But I'll do my best to try and talk about some of the key issues that I think are facing public health and the opportunities that are also facing healthcare systems from a public health perspective. So this is a slide that I got from Mike Standing at Deloitte, and of course, because it comes from the consulting agencies, it's mostly hand-waving, but it is a sort of interesting thought. And that is, if you look at the way we spend money in healthcare spending in the US today, it's mostly around symptomatic and disease-modifying therapy. And there's almost no money, particularly in the US, spent on curative or preventative interventions. And they've kind of been missed as part of that agenda. But the speculation is, as we go 20 years out, that's going to change quite dramatically. And in fact, I think it's pretty evident that it has to change, because most healthcare systems at the moment are at the point of breaking, largely because we don't do enough curative and preventative interventions. So I think I just put this up not because I think it's very factual, but because I think it does make you think about how healthcare systems are going to adapt. And I think on the whole, industry as well as academia needs to be involved in this shift in healthcare spending. So the way you approach public health, of course, goes all the way back to John Snow and the Broad Street Pump, but also to this guy, Jeffrey Rose, who I think was the first to really argue that actually the right way to approach public health interventions is to shift curves. So to shift curves to the left to reduce risk in whole populations. And that's the best way of reducing risk, rather than selecting with precision the high-risk population and doing something in them. And the impact of that, he argued, is a better impact. The problem with this is it basically hasn't worked. And the reasons for that are, it has worked in some settings, so clean water, clean air, those sort of things. Things where state intervention can be used to systematically change the way things work. And also situations where you can get very high levels of compliance, which, as you know, is pretty hard to do in any setting. So it turns out that this methodology is not very cost effective, because you're spending money across a whole population, including on a lot of people who are never going to get the disease that you're interested in. And there's a big issue of compliance as well. So it's not really always cost effective, and it's often pretty inefficient. And I think a good example of this is behavioral change modalities, which have, I think all would admit, been pretty unsuccessful in terms of managing the major chronic diseases. And that's because they've been blasted out at the whole population, and that largely hasn't worked. So I guess one domain where you might say this works, but we're in the middle of testing this, is whether population-wide shifting the curve stuff is going to work for infection control. And we're right in the middle of a really interesting experiment. And my bet, based on the Italian experience, is it isn't going to work. So I think there are lots of reasons why the model of shifting the curve works in some circumstances, but in many circumstances it isn't going to play. And of course, Mike Richards' report on the independent review of adult screening program, which again are a very good example of where we screen across whole populations, was pretty negative about the benefits given the resource put into large-scale cancer screening programs, largely because you are screening you're screening basically on the basis of age rather than risk of disease. And he made the argument in this report that we need to be more precise in thinking about how we apply screening programs. So we also live in a world where we've got a lot of new tools for public health. So public health has really been transformed in the past decade. First of all, we can now analyze risk determinants in a much more effective way at scale across populations, and they are not just genetic and genomic risks, but also behavioral risks and environmental risks. And it's the combination of those three things which really gives a risk profile in any given individual. And it's possible now to do that at scale. And the tools that are available for doing that, of course, are the drivers for our ability now to look more precisely at risk. And that's in large part due to the digital revolution, which is giving us insights into types of data that we never had before. But also new types of diagnostic tools. The world of diagnostics has moved on enormously and is allowing us to interrogate wide sets of different biomarkers, and of course genomic data, which has now been brought to the point that it can be used across whole populations. And if you back those risk assessment tools into the interventions that are available, we've also got, I think, a range of opportunities in interventions that will change the public health arena dramatically. One is behavioral change, and the ability, I think what's evident is the failure of a variety of techniques to change behavioral change, I think, are pretty evident across the whole public health space. But the people who do know how to change people's behavior are the digital companies, and I think there's an interesting question about whether their abilities can be aligned with public health initiatives to try and be more effective in changing behavior. And a wide range of pharmaceutical interventions, both for protection and for early therapy, are now increasingly available. So I think there are a host of new tools that we've got to actually be much more effective at reducing an individual's risk and diagnosing diseases early than we had before. And finally, and this is also important, and that is one of the problems with public health studies is that they often take a long time and they require very, very large numbers of people. And this is true actually with intervention studies for most of the major chronic diseases, and that is they basically price themselves out of the market. And that's now changed because digital tools are now available to allow you to do that at scale. So the world looks really different than it even did 10 years ago for public health and particularly for precision public health. And these are the three main domains of risk that you see in any individual for a whole wide range of diseases. And they're all now much more quantifiable. Environment is still hard because it's really area under the curve across a life course, so that's quite difficult still to measure, but it's not without its successes. Genetics, as I've mentioned, has become scalable. And interestingly, we've got many more data points on social environment, but also behavioral factors. Now, whether you can actually get at the behavioral data, it's not clear, but if you had access, for example, to the Facebook data set, you would have an enormous amount of behavioral data that would tell you lots of things about lots of people, and which could be then correlated with disease risk and disease outcomes. So I think all these tools all have been enabled by many of the changes in the modern world. So that gets us to this issue about can you make public health more precise? And the philosophy here is pretty simple, and that is one of the reasons that the healthcare system here and elsewhere doesn't work very well is that we tend to focus a lot of resources on a lot of people who actually will never get sick and will never get the disease. And that's not a very efficient way to operate. And if you look at examples of public health interventions that have had major success, probably the best is cardiovascular disease, where by measuring a few biomarkers, blood pressure and LDL cholesterol, you can target people who are at particular risk, and you can really substantially reduce cardiovascular outcomes. In the last 30 years, they've fallen about 70% in the UK. And so that is a good example of how targeting subpopulations can be hugely effective. But this whole issue is also very relevant to the other bit of the agenda, and that is the natural history of disease starts with people who have a particular risk portfolio, but then they often spend a very long time in the prodromal periods of their disease. Cancer, for example, probably at least a decade for most cancers. Living with cancers, cardiovascular disease probably starts in our 30s. Obesity probably starts shortly after birth. There's a lot of long prodromal periods where there will be real opportunities to intervene. So the early detection agenda becomes very important in this space. And you may also be able to improve the effects of things like behavioral change modifications by targeting individuals who are at particular high risk of disease, or where you think the behavioral modifications are likely to be tractable, because it should be possible to measure and evaluate who will be responsive to behavioral change and who shouldn't be. So this is the new world, which I think is now rapidly coming on us, and I think it's a really interesting opportunity. So you may wonder why Munir asked me to come, because I don't really do public health, but I did help with this, which was the Life Sciences Industrial Strategy for the government, which we published 2.5 years ago. And what was interesting is that when you talk to industry about where the future of their business is, many of them will describe things that really relate to large-scale interventions in populations that could be described as public health. Now, they would never describe it as public health, because it's well known you never make any money out of public health. But the truth is, what a lot of people are now thinking about are really public health interventions, which are interventions at scale to try and prevent or intervene early in the major chronic disease populations. And the three main, one of the things we described in the report was to try and identify 3 new life sciences industries that the UK could develop over the next 20 years. And the 3 were genomics, digital health, and early diagnosis. And you can see that actually, based on the comments I've already made, all these do intersect with the public health agenda. These are all public health initiatives that will change the face of how we define disease, how we intervene, and more importantly, when we intervene in those disorders. And they're all domains where the UK has a very particular advantage. Obviously, in genomics, we've got enormous advantages in sequencing technology, precision medicine, as illustrated by today's meeting, target discovery, genotype, phenotype in public health. The digital health agenda, which you're all familiar with, also will enable all these things. And then this whole domain of early diagnosis, which I'll come to in a few minutes. So let me first of all start with genomics, because I think in the last 3 or 4 years there's been quite a dramatic shift in our understanding of the way genomics could impact on healthcare. And as you know, there has been interest for a long time on the involvement of rare variants with relatively high penetrance in producing diseases, largely rare diseases, Mendelian diseases in populations. And of course, the UK in many ways leads the world in this space, because we've analyzed very large numbers of Mendelian diseases in Genomics England. This is the BRCA1, mutations of BRCA1 on here, but there are many others. And the penetrance of these tend to be pretty high, and they're interesting, but they're rare. So on a public health level, they're interesting, but not hugely impactful. So what has emerged, though, is that common variants, when aggregated around individual disease to produce polygenic risk scores, do provide really pretty interesting levels of risk in population that will allow you to identify high-risk populations, but based on the aggregated risk from their polygenic risks around any one of 20 or 25 different common diseases. And it'll probably be true that these are relevant for a whole wide range of phenotypes, not just disease, but also response to therapy, adverse effects, there's no reason to believe that polygenic risk scores won't be relevant in all those domains. Now, some of you will recognize this picture. This comes from the NIH. And when this started, there were only a few dots on these chromosomes. Each one of those little blurs is a dot relating to a locus related to a disease. So there are literally tens of thousands of these variants which are now available that can be aggregated around any single disease. And that, I think, is where genomics is most likely to impact on the public health agenda, because it'll allow us to identify the populations at risk. And having said that, the UK has positioned itself really powerfully around this wider genomic agenda. Genomics England is doing largely rare variants in rare disease and has started on a cancer program, although not yet finished getting that right. But that's a great asset for the UK to have. We've started a project sequencing the whole genomes of all 500,000 people in UK Biobank. That project is now 9 months in. They've finished, I think, 160,000 sequences. So they're well on their way of getting those done, and they'll be completed, those 500,000 sequences, in 2 years. So that'll be the largest single sequencing project on the planet, and will be crucial for the functional genomics that will follow. And then, thirdly, we are also having a big pitch at early diagnosis using polygenic risk scores across a large population of people, and I'll come back and talk to that in a few minutes. And behind that, there is a national genomics strategy, which hasn't been announced yet for a whole variety of complicated political reasons, but again, this describes the 3 domains that we're pursuing in the UK: clinical genomics with Genomics England, UK Biobank, which is really focused on genotype-phenotype relationships, and then this public health genomics domain, which is around the large 5 million patient cohort. So alongside that, in the second bin, as it were, is digital, and it's not a separate bin because genomics is a very important bit of this, but there's a whole load of stuff that falls into that, and that is going to allow us to identify patient populations much more precisely, but also be able to use that data to do the kind of studies that have been very difficult to do in public health, because they're long studies that involve tens of thousands of people where you have to measure outcomes over a long period of time. And they have proved to be extraordinarily expensive, difficult to do, and that is now changing because of the digital environment we live in. And the UK's digital environment is pretty impressive and is getting steadily better. And this is a sort of summation of what that looks like. So data assets across the devolves as well as England is about 65 million people. We will get access, I think, by the end of this year to complete GP records, where there's 15-year legacy data on all people, which will be mostly the structured data there, which is lab data, prescription records, hospital discharge data. So that's important. We already have coded hospital admission data and outpatient data. There's quite a lot of pathology data in the spine, but interestingly, with the new program that we launched through the Life Sciences Strategy for Digital Pathology, we'll soon have 30 million people covered with digital pathology images on all their pathology, which is by far the biggest in the world. So that's another piece that works really well. We've got lots of large-scale cohorts, UK Biobank being the most famous. A lot of registries which don't work very well, and there's an effort now from government to fund into trying to make those registries both accessible and more real-time. We expect to see genomic enrichment in 5 million people and networks for digital pathology and radiology. And then that's backed up with a whole variety of other structures. So globally, the UK is in a really unique position to do this. And you don't need much imagination to understand how that data will allow us to identify populations that will benefit from public health interventions. And then the other piece from data comes from all the technology that's available. Some of them I've just mentioned, but there's, of course, a whole load of other things, wearables, imaging, and a whole variety of other tools. And the aggregation of that data is going to give us a much more precise understanding of an individual's particular risk. but also which subset of the population is at particular risk of particular diseases. And that's actually quite a game changer and will, I think, have a pretty big impact on the whole field of public health. Now, one of the things which I've alluded to is that there have been constraints in doing large-scale studies, and in fact, it's got substantially worse. So in about 1990, the IHGCP rules were developed for for trials of all kinds. And the idea was a good one, and that was to try and make sure a trial done in Japan was the same as a trial done in the UK. And it was a good idea, and there were standards imposed. And because there were standards imposed, most pharmaceutical companies turned the responsibility over to CROs to do this. And of course, the CROs, as they should have, saw this as a massive commercial opportunity, so they just basically got a hold of the tap and turned it on steadily over the past 30 years. It's also worth remembering that in 1992, when this was introduced, no one had a computer. So it wasn't possible to think of any way of doing this except very heavy lifting by sending clinical trial monitors out with clipboards, pencils, and tick sheets. So that's basically why we ended up in this muddle. Now, the CRO industry is now an $80 billion industry globally, and it's the main reason why you can't do large-scale studies. So a large-scale pivotal study in cardiovascular disease prevention of, say, 40,000 patients will cost you close to $1 billion. So guess what? Nobody does those studies anymore. And the drugs that might be accessible to us for these very important indications are not being made accessible because pharma, who are not stupid, have migrated to specialist pharma, rare disease, highly specific indications where they can get their trials done for a much lower amount of money and they can be commercially successful because people get paid more and it's all fine. So the problem that leaves is that nobody's working on the major causes of morbidity in healthcare systems. Cardiovascular disease, diabetes, obesity, some of the major cancers, but chronic respiratory ailments and the like. So this is a massive problem, and it also makes conventional preventative interventions very difficult to quantitate. So the reality is that GCP has probably outlived its usefulness. Pharma has moved to specialty pharma. There are very few new interventions, and most big pharma companies have moved out of this space, so they're not producing there anymore. And so the goal of some of this infrastructure around data must be to get back where we reduce the cost of testing these interventions by increasing the efficiency of those trials. And that's one thing that we've been involved in coming out of the life sciences strategy, and that's to try and digitize both recruitment and data capture in healthcare systems to revise the GCP guidelines, and the Wellcome and the Gates Foundation are running a rather helpful working group on that at the moment. And we're now currently running an exemplar using NHS digital data to reduce the cost of those studies. And I'll just tell you a little bit about that because it relates to a new therapeutic modality, which is siRNA. This is, again, a major focus in the UK at the moment, is to make sure that, unlike antibodies, which we invented and then completely missed the therapeutic opportunity, we don't want to miss the therapeutic opportunity associated with gene and nucleic acid-based therapies. So there's quite a lot of stuff going on in that space. We've got a lot of success in this space. Nightstar was bought, Orchard's independent company, Oxford Biomedica's a great company. So we've got a lot of play. But we were approached by the Medicines Company, which is a Boston mid-sized biotech company, with a siRNA to PCSK9, which, if you inhibit it, reduces LDL cholesterol very powerfully. And they approached us largely because they had early data, now completed all their phase 2 studies, that show that with an injection, certainly a 6-month injection of this PCSK9 inhibitor, you could reduce LDL cholesterol by about 50%. And it also looks like the data based on annual injections is almost as powerful. So this is a classic example of a public health tool, because you could use this to drive compliance of these of such a therapy. And this is basically the Orion 11 study, which used this small interfering double-stranded RNA, and it interferes with the transcription of PCSK9. It gets into the liver well with GalNAc conjugation, which makes it an ideal therapeutic intervention. And the news of this phase 2 stuff came out right at the time we were all getting depressed by all the newspaper headings, which were over here. This was back in September. Do you remember all this? It was all a bloody disaster. But anyway, the good news was that we had a new public health intervention for cardiovascular disease protection. And this is the data that came out of the Orion 11 study, which was the phase 2 study. And you can see that this is really quite a profound reduction in LDL cholesterol. And the advantage of the injectable is that the data shows that if you put people on a statin because of elevated levels of LDL cholesterol, almost all of them stop taking them at 5 years. But this will allow you to avoid that. So I think that, and those studies, we're now doing 15,000 patient secondary prevention study and a 40,000 patient primary prevention study, and we're doing them at about 10% the cost that we would do if you were doing it with a CRO. So that could dramatically change our ability to do those kinds of studies. So the final thing that came out of the life sciences strategy, which is very relevant to this agenda, is this ADD initiative. And just to be crystal clear, we're not going to call it the ADD initiative. We've got to go back and think of a new name for all the obvious reasons. That's a name that came from UKRI, so that's not so clever. But anyway, we will call it something else in the end. But this is really to address this issue of early diagnosis and early identification of high-risk populations across all the major chronic diseases: cancer, cardiovascular disease, inflammatory diseases, neurodegenerative diseases, and the likes. And it should be possible now, with a set of the tools I described earlier, to identify people at high risk of these disorders, and then see whether you can use tools to identify those with very early stage disease, or even just those at the very highest risk. So we can use public health interventions in those that may be things like behavioral change and the likes. So, and this has all been enabled by a remarkable revolution in new diagnostic technologies across many diseases that range from different types of imaging, circulating tumor DNA, biomarkers for Alzheimer's disease, lots of possibilities. And the idea was to, instead of doing multiple smaller cohorts, to do a single big cohort across the population. The reasons for that are shown here, and that is that most of these chronic diseases have a long pre-symptomatic period. You can see this is the impact of statins in a population where you intervene in a high-risk population based on that. And the cancer data we're all familiar with, and that is diagnosing cancer at an early stage is probably by far the best way of improving long-term survival. So the study that we're doing, and we just hired a chief executive who starts in 2 weeks' time, is to collect 5 million healthy individuals, participants from the ages of 30. Actually, there's no top limit. We'll take anybody into the study if they want to participate. They're consented, and they provide their NHS health records. We do polygenic risk scores in all of them. We get access to the NHS patient record. We will save plasma and DNA in all 5 million, and in 1 million we'll do repeated blood samples on a yearly basis. And then that cohort can be used by all kinds of scientists to do highly intensive studies on people who are at high risk of esophageal cancer, colorectal cancer, cardiovascular disease, and the likes. So it's actually a rather important piece of infrastructure if you're going to try and do these projects at scale in the UK using NHS data. It will allow us to test a whole load of new tools and measure outcomes quantitatively. Of course, we've got a great record in cohort studies, and the intention here is to try and build a cohort which is big enough to get sub-cohorts at scale, and as a result, we've had to increase the size substantially. The availability of polygenic risk scores will help in defining risk, but it's not the only thing. There are lots of other ways to define risk in populations, including behavioral risk and environmental risk. And it will be very much a research study, but one that overlaps with the healthcare system. And we've got great support from all the big life sciences companies, the charities, and government that will allow this to happen, and the pilots are about to get launched. Now, within this, we'll also be able to do other studies. There are a lot of people interested in using digital tools, to recruit the study, because that is a mechanism to get 5 million people engaged, is to get lots of people to download the app. to consent on the app, and to be reapproached with data from the app. So the intention is to go back and forth, and also to do behavioral modification using digital tools. Now, there's just a couple of things that this have raised, and then I'm going to stop talking because that's it. And that is, in many ways, having thought about this, one of the things that emerges is that the biomedical community has been captured by ROC curves, which are a good way of measuring sensitivity and specificity around a single test. And of course, we all know about those, get taught as medical students, that that's the way it works. The truth is, that's actually not how doctors make diagnoses. Doctors make diagnoses based on a much more Bayesian approach. So they go to the bedside and they see a patient, and the patient's thin, is a bit jaundiced, maybe looks a bit anemic, and they go based on that, using that as a prior to make decisions about what they're next going to test. And I think there's a new paradigm for diagnostics that will be very important for early diagnostics that actually says you start with the risk, because you can be much better at defining risk in patient populations. And then you use biomarkers, maybe ctDNA, to get a much better read as to which of the subpopulations. And then, ultimately in the cancer setting, you'll do imaging to decide who's got what cancer. And I think we need to move away from the classical view that you take one test, you do an RC curve, and then you throw it in the bin or you keep it. Because, the truth is that's never been the way that medicine actually works. And it's much more of this. I mean, this is just an example, but of course there are many other examples of how we use a Bayesian approach to that. I'm not going to spend much time. Most of you know about polygenic risk scores, and you're going to get a lot more today. But I think one of the interesting things about this is you do get some pretty eye-watering elevations in risk in populations, depending on how far to the right on the curve you want to go. So, if you want to go to the top 3% of risk, for example, in a large population, which, to be clear, in the ADD cohort will be 150,000 people. That's a massive risk in populations that will allow you to identify people who are at very high risk of these diseases. So that is a very important, but not the only piece of the risk portfolio. And this is work done by Peter Donnelly, of course, has led this field since he did the original Wellcome case control study. But this just basically shows what happens. You start with the conventional risk for cardiovascular disease, and you add the PRS. From the PRS, there's quite a lot of people who were at risk who no longer are at risk. And there are also a lot of people who were not at risk who become at risk. So you actually change quite dramatically the way you parse patient populations into the risk and non-risk population. This you will have all seen. This is the obesity story published in Cell last year. And this is the difference in weight in kgs in people who have either from the high polygenic risk ratio to the low polygenic risk ratio. And you can see the differences are just marginally there at birth, but they're clearly there at the age of 8, and then by the age of 18, you're done. So it does show the power of genetics in defining that risk. This is just some pragmatics about how we're going to collect the study, and this idea of using digital tools to nudging people who are at particular risk, I think, is a pretty interesting thing, unproved to at this time, but a massive opportunity in these sort of populations. So that's the ADD project. These are the reasons we're going to do it, but I think I should stop there, and thanks very much for listening.